So this video is of the genetic series, and it is video number 8 of the genetic series, and this is about triplet repeat expansion disorder. So normally some of the gene have triplets, and they have repeated triplet. So if there is expansion of the triplet repeat, what happened that there is either the gene product such as protein synthesis is decreased, like in case of Fragile X, or in case of Huntington disease due to the triple repeat expansion, the product of the gene become neurotoxic, specifically to the basal ganglion that produce the abnormal movements. So normally we have these terms that could be important. So for instance, let's take an example of uh, FMR1 gene for the Fragile X. What happened that it normally has some CGG repeats, but if these repeats raises up to 59 to 200, we call a pre-mutation has occurred, and in such individuals we see certain manifestation. For example, in a male Fragile X, associated tremor ataxia can be seen if he is pre-mutated, such as the CGG repeats number is somewhere between 59 to 200. In female, we see fragile associated premature ovarian failure. And in meiosis during gametogenesis, these CGG repeats may increase the number of 200, which is, and we call a full mutation has outcome, and so it can be transmitted to offspring with full mutation, and no, actually in the case of Fragile X, no protein synthesis. So for this, we observe another phenomena that is called anticipation. So in the CGG repeat expansion, we see that if the disease is occurring with more severity in the next generation, and it also occur with at early age onset. For instance, if the mother of an individual with Huntington's disease was affected somewhere in her 30, 30s or 40s with slight movement disorder, the daughter or the son will be affected somewhere in 20s and 30s, and he will be having severe motor disorder. So we call it anticipation, and it is due to increased trinucleotide repeats. And we have certain examples, like for example in Fragile X is CGG repeat, Huntington disease we have CAG repeats, and myotonic dystrophy we have CTG repeat, and Friedreich ataxia we have GAA repeat. So these are the conditions that we see normally with trinucleotide X repeat, triplet repeat expansion disorder, and these are actually trinucleotide repeats. Thank you so much.